Hello, Lana. Welcome to our first lesson. We want to look at the definition of the cell, structure and function of parts of a light microscope. So, the cell is the basic unit of an organism. And all organisms are made up of cells which cannot be seen with the naked eye. Therefore, the aid of a microscope is required. What is a microscope? The microscope is an instrument used to magnify or enlarge objects. The most common type of microscope is the light microscope that uses a beam of light to illuminate the specimen being studied. This light microscope is the most commonly used microscope in schools and institutions that do not focus on very fine details of internal structures of cells. So let us look at the history of the development of light microscope. So in the year 1650, a man by the name Zacharias Janssen invented the compound microscope, which combines two lenses for greater magnification. In 1665, Robert Hooke used an improved compound microscope to observe cells, or precisely the Koch cells. Between 1650 and 1700, Anthony van Leeuwenhoek developed a better microscope with lenses to provide a greater magnification. He used the microscope to view nuclei and unicellular organisms, including bacteria. The development of the electron microscope in 1930s significantly improved microbial studies. Through this microscope, it was possible to study finer details of structures. So, this is the structure of a microscope. So, we have the eyepiece legs, from where you will just observe something on the stage. And then we have the body tube here. We have the revolving nose piece. And then we have the objective lens. There is the arm. Then we have the stage. On the stage, we have clips that will help in fixing or holding the slides in position. And then we have the coarse adjustment knob as well as the fine adjustment knob. And then there is the base or the stand. We have the diaphragm and of course the mirror. So that is the parts of a microscope. So let us look at the functions of parts of the light microscope since we have seen the structure. So on the first, we have the limb or the arm. This part supports the body tube and the stage. We have the base, which provides firm and steady support to the microscope. The body tube holds the eyepiece and the revolving nose piece. We also have the coarse adjustment knob, whose function is to raise or lower the body tube through longer distance to bring the image into a rough focus. For the fine adjustment knob, it raises or lowers the body tube through smaller distances to bring the image into sharper focus. It is mostly used with high power objective lens. The role objective lens is to bring the image into focus and magnify it. The diaphragm is an aperture regulating the amount of light passing through the condenser to illuminate the specimen. Apart from that, we have the eyepiece lens, which contains lens that contribute to the magnification of the specimen under review. The mirror reflects light through the condenser to the object on the stage. The revolving nose piece holds the objective lens in position and also enables change from one objective lens to the other. For the objective lens, we have three types. We have the low power objective lens, the high power objective lens, and the medium power objective lens. So when we are viewing specimen on the stage, we begin with the low power, then the medium power, and then the high power objective lens. So it is possible to change those powers through the use of revolving nose piece. We also have the condenser that concentrates lights on the object on the stage. And the stage is that flat platform where specimen on the slide is placed, and it has two clips to hold the slide in position. So we want to move ahead and look at handling and care of the microscope. So in our schools, how do we care for the microscope? So we should always use both hands when carrying the microscope. That is, one hand should hold the base to provide maximum support, as the other hand holds the limb or the arm. Never place the microscope too close to the edge of the working bench or a table, because it is likely or easily to fall down and break. Never touch the mirror or the lenses with your fingers. The dirty lenses should be cleaned using a special soft lens tissue paper or tissue paper moistened with ethanol. The other parts of the microscope may be cleaned using a soft cloth or tissue paper. And do not wet any part of the microscope, as this can cause rusting. Always ensure the low power objective lens clicks into position in line with the eyepiece before and after use. After use, always clean and store the microscope in a safe place, free from moisture. As we have said, moisture will make the microscope parts rust, and dust will make it dirty, will dirtify the lenses and the mirror. We move to how to use the microscope. So if we have a slide to prepare, or we have prepared a slide and we want to observe so that we can be able to draw and describe, what are we supposed to do? We have to place the microscope on the bench and ensure the stage is facing away from you. Then turn the low power objective lens until it clicks into position. And what will help you do this? It is the revolving nose piece. Ensure that the diaphragm is fully open so that light can pass to the stage. Look through the eyepiece with one eye. Meanwhile, adjust the mirror under the stage to ensure that maximum light passes through. So, and as you are doing this, you move next to a window or a place where there is enough light. So the circular area you will see is called the field of view. Place the slide containing the specimen on the stage and clip or fix it into position and ensure it is in the center of field of view. Again, look through the eyepiece while adjusting the mirror under the stage to ensure that enough light is passing through the specimen to the specimen. Use the coarse adjustment knob to bring the low power objective lens to the lowest point. Viewing through the eyepiece, turn the coarse adjustment knob gently until the specimen comes into focus. Then, for us to have a sharper or a better image, use the fine adjustment knob to bring the image into sharp focus. That is a very clear image. Then, make a drawing of what you observe. If higher magnifications or finer details are required, then you switch from the low power objective lens then to the medium power objective lens and adjust to the focus using the coarse adjustment knob. And for sharper images, you use the fine adjustment knob. And if more finer details are required, you turn to the high power objective lens and then use the fine adjustment knob to bring the image into sharper focus. The next thing we want to discuss is magnification. So, the magnification power is usually written on the lens. For example, from the low power objective lens, we have that is having times 10, maybe the medium power times 40, others times 100. So that is when it is written times, it means the number of times the image or the number of times that lens is able to magnify. So to find out how many times a specimen is magnified, the magnification power of the objective lens is multiplied by that of the eyepiece lens. For instance, if the magnification on the eyepiece is times 10, that means the eyepiece has the ability to enlarge 10 times. And the objective lens is able to enlarge 4 times. Then the total magnification is times 40. And remember, earlier we had said that magnification has no units. Example, if an animal cell viewed under a microscope was magnified 675 times using an eyepiece lens of magnification power times 15, what was the objective lens power? It's very simple. Let us look at the solution. So since we've said magnification is calculated by multiplying the eyepiece lens power by the objective lens power, then we shall find the objective lens power by dividing the total magnification over the eyepiece lens power. That is 607 times 675 divided by times 15 to give us times 45. Example 2. Complete this table using this formula. For example, if the eyepiece power is times 5 and that objective lens is times 4, that means the total is 20. So you will be able to calculate and fill this one. And for you to find the eyepiece lens, you will just have to substitute and divide the total magnification power by 10 and you will get 10. Very simple as that. So we are through with our first lesson, but here is work for you. Make sure you attempt.